Thank you everybody for joining us today for the Everything ALS bi-monthly webinars, where we love bringing the experts to your living room. My name is Lisa Deegan. I am part of the Everything ALS team because I lost my younger brother, John, to ALS a few years ago. So our aim is basically to provide everybody with what we didn't have throughout our journeys and to help further research in ALS through our digital biomarker speech study, so we can help put an end to ALS. Today, we want to welcome Dr. Walter G. Bradley. He's a renowned expert in ALS and deeply involved in research to find its cause and its cure. He specifically has been researching the interaction of environmental factors with genetic and temporal factors in the genesis of ALS. Dr. Bradley is a native of England, and he has practiced both in the US healthcare system and in the British National Health Service. He served as chairman of the Departments of Neurology in Vermont and the University of Miami for 25 years. He's a professor and chairman emeritus at the Department of Neurology at the University of Miami. So I want to thank the audience again for tuning in, and a big warm welcome to Dr. Bradley for being here tonight to share your research findings with our community. Thank you. The floor is yours.
Lisa, ladies and gentlemen, thank you very much indeed for being here, and thank you for listening to me. I'll try to keep within something like a reasonable time so that you can ask questions afterwards. But what I'm going to try to do, if I can actually work out this technology, is to share my screen and show you some slides. So Lisa, thank you very much for the introduction. As she said, I've been looking after ALS patients for more than 50 years, and in fact have been doing research particularly into environmental causes and more recently genetic causes of ALS for the last 15 years, something of that nature. So I want to try to help all of you understand where I think we stand at this moment with regard to understanding ALS. So ALS is in fact known in Britain and the British Empire as motor neuron disease. In the French colonies, it's known as maladie de Charcot. It is in fact not an unusual disease, though in fact I heard some comments earlier about never having heard of it. When somebody is diagnosed with it, they in fact find out that they've several friends or neighbors have in fact had it in their area of their life and so forth. So it is much more common. If you're age 50 and above, the lifetime risk of developing ALS is 1 in 100, and that's not a rare disease. It in fact is not a difficult diagnosis, and in fact many patients come to the doctor having already made the diagnosis for themselves from Google. But in fact, it can often be made, the diagnosis, without special tests, but nevertheless, no doctor likes to give the diagnosis because it has such a poor prognosis. And so in the US, certainly, neurologists often don't tell patients that the diagnosis, and spend up to 12 months doing test after test before eventually sending the patient to an ALS center to be given the diagnosis. And that's a terribly difficult time for all of those patients. So the diagnostic characteristics of ALS is a slowly progressive worsening of upper and lower motor neuron degeneration of adult life. Now, the upper motor neuron problem leads to spasticity, stiffness. The lower motor neuron problem leads to muscle wasting. The age is generally above the age of 35, with a peak incidence of around about 65. Males exceed females by about 1.5 to 1. And the average survival from diagnosis to termination is between 2 and 5 years. But there is a wide range of people who survive much longer than that. I've known patients who have survived for 40 years and are still working. And there are a small number of cases that burn out or seem partially to recover. And again, I've seen 5 such patients over my career. So that's always something to be hoped for, but sadly not very often seen. So classically, there are no extra motor problems, but in fact there are ALS plus cases that have a number of different disorders, the most common being what's called frontotemporal dementia. So these are the first hints that ALS is not a single disease. There are various types of ALS. There is the classic ALS that has what I've just described as being both upper and lower motor neuron signs. It can start in any part of the body, the arm, the leg, the swallowing mechanisms, speech mechanisms, respiratory, but then spread to other areas of the body. But there are other forms that really remain more or less isolated. For instance, primary muscular atrophy, primary lateral sclerosis, and so forth. And then, as I said, there are these variety of different cases that have an overlap between Parkinson's disease and ALS, ALS and dementia, and so forth. So those are the things that I think make us begin to wonder whether we really are dealing with a single disease. And I'll talk to you a bit more as we go forward about that. So, as I've said, ALS is a syndrome with many causes. About 10% of the cases are familial. There is an underlying gene in those families. About 50% or 60% of those genes have now been identified. And those research into those genes has helped us understand some of the processes that lead to motor neuron degeneration. But in fact, 90% of cases have no family history. And in those, what are called sporadic ALS cases, there have been identified both a number of environmental risk factors, which I'll go over in a minute, and a number of different genetic underlying predisposing factors. And that combination has led to the concept of gene, environment, and time interactions. And the time is because it takes a long time of exposure, and we know that for sure from a number of different diseases, cancer, these neurological degenerative diseases and so forth. It takes a long time of exposure before people begin to develop the disease. We're talking about 10, 20, 30 years, and even exposure at the time of birth. So let me now turn to the environmental epidemiological research that we've been undertaking, which has been certainly fascinating, interesting, and has been a real quest, which originally started in Guam. And I'm sure all of you have read about this, but let me go over this a bit. So Guam is an island in the Mariana Islands in the Central Pacific. And in Guam, these islands, in fact, were captured by the Japanese in the Second World War. And when the British, sorry, when the Americans got back into Guam at the end of the Second World War, they found that the risk, that the number of ALS patients, they had never seen this number of ALS patients. About 25% of the deaths in Guam were due to ALS, and the frequency of the disease in the island was about 200 per 100,000 of the population, which is about 100 times what it is in the rest of the world. And obviously there had to be a local cause of it. There are a couple of other foci around the Pacific, one in the Key Peninsula in Japan and the other in West New Guinea. And a lot of research was undertaken in the next 20 years trying to find what was the cause of that environmental factor that was responsible for the disease. Interestingly, over the next 20 to 30 years, gradually the frequency of ALS decreased to about currently around about 5 per 100,000 per year, which is still above the incidence in the rest of the world, but is certainly not what it was when the American troops got into there. And in fact, there's been also a shift of the type of disease, with more of the patients showing Parkinsonism and dementia. So a lot of research, as I said, went into trying to find what was the cause of the disease. The cycad tree and its fruit, its seeds, immediately fell under suspicion, because the Chamorros used those seeds for making their tortillas, and a lot of the animals that they eat feed on the seeds of the cycad tree. There is quite good epidemiological evidence that, in fact, the exposure to the whatever it is in the cycad seeds has to be of long term, and the evidence for this is listed on this slide. When Chamorros emigrated to California, they carried on having this very high risk of developing ALS for more than 10 years after they'd left the island and left the way of life of the Chamorros. And similarly, when people from the Philippines emigrated to Guam and adopted the native lifestyle, then they did not start to develop ALS for at least another 10 years. So both the onset and the washing out period were really quite prolonged. And it bears out what I said about this being a very chronic neurotoxin that must be responsible. Now, in the early stages of the research in Guam, scientists went looking for these toxins, and they found a large number of toxins, which the predominant ones among them are acute toxins, toxins that cause liver cancer, liver cirrhosis, mutations in cancer elsewhere, and so forth. But really, the evidence did not link up to suggest that any of these were related to ALS. Now, in 1967, Bell and colleagues discovered a non-protein amino acid called BMAA that they thought and were able to show was neurotoxic, and they thought it might be the cause of Guam ALS PDC. Now, a non-protein amino acid means that it's an amino acid, but not one of the 20 amino acids as in our proteins, because it has a variety of different chemical links added to it. So here is BMAA, which is comprised of alanine with a chemical group called methylamino linked to it, which is why it's called beta-methyl amino L-alanine, BMAA. Now, the BMAA story was carried forward by Paul Cox, whose picture 20 years ago is on the right-hand side of this. Paul is a wonderful research worker who has made it his life's work to try to chase the causes of ALS. And in 2002, 2004, they were able to find that there are cyanobacteria, and I'll show you those in a minute, in the Chloroid roots, which are roots of the cycad tree that get above the ground and are used by the cycad tree to fix nitrogen, that is to make fertilizer for the tree, and to also make other substances, including some, unfortunately, of these various neurotoxins. So Paul and his colleagues were able to take that story all the way through to looking at the BMAA in the proteins of the flying foxes, the animals that eat the seeds, and eventually into the brains of the patients who suffer from ALS. So here are the Chloroid roots. They're really quite, they're quite small. And when you see them, this is a nickel, obviously, and here are there's the Chloroid roots cut across, showing this green ring inside them. And that green ring is, in fact, cyanobacteria. And these are the cyanobacteria here. And they have what's called a symbiotic relationship between cycad and this cyanobacteria, the this called nostoc. So here, flying foxes eating the seed of the cycad. Here's the pathway of it getting into the brain of patients who die of eventually ALS in Guam. Now, cyanobacteria are ubiquitous, that is, they're everywhere in the world. These are Hawaiian Islands in the Pacific, and this is a great big massive bloom of cyanobacteria there. This is Florida, and here's a massive green collection of cyanobacteria. This, in fact, is one of the guacamole-like glob of cyanobacterial bloom that we get down here in Florida every few years. This is a lake in New Hampshire that has cyanobacteria in it. And here in the Middle East, in Qatar, here's Paul Cox and I finding that there are cyanobacteria in the what are called the cryptogamic crust in the desert. You'd never think the cyanobacteria, which is a waterborne organism, would be actually in the desert. But what happens is the rain comes about twice a year. It fills these depressions. The cyanobacteria reproduce like crazy. They die off fairly quickly and form spores and go back resting until the next rainstorm. So cyanobacteria everywhere. And we thought that possibly, therefore, they were the main causes of ALS. And I think we've now come to realize that that's not true, but work has certainly been done to tie up the linkage between BMAA and neurodegenerative diseases. This is a paper where BMAA was fed to vervet monkeys and chronically produced the changes of Alzheimer's disease in the brains of those monkeys. And we went on and showed that the changes in the brain were also mimicked in the spinal cord and were very similar to ALS, and further showed that, in fact, L-serine, one of the normal amino acids that is in all of our diets, can, in fact, if given in large doses, can reduce and partially prevent the development of these changes of ALS and Alzheimer's disease. So, but cyanobacteria, as it turns out, are not the only cause of ALS. And I think we would have been foolhardy if we believed that that was the case. So there are a number of other environmental and lifestyle factors that have been linked to sporadic ALS. The exposure to them leads to a greater frequency of ALS in a population compared with a control population that are not exposed to them. So, for instance, we've talked about cyanobacteria, but exposure to lead, to pesticides, to mercury, having repeated electroshocks. Smokers have a higher risk of developing ALS. Those in the military who are deployed have a higher risk of developing ALS. Some sports, particularly those associated with head injuries, have a higher frequency of ALS. And a number of occupations working with solvents and agriculture have also a higher risk of developing ALS. So there are multiple environmental factors that are linked to the frequency of developing ALS. And this has always interested me. The best demonstrated relationship between head injuries and ALS, which had been known for a good number of years, boxers, American football players getting repeated concussions and so forth, developing ALS. Lou Gehrig was the first of these, and that's why it's called Lou Gehrig's disease, obviously. So a sportsman and probably a number of repeated head injuries, and he goes and gets the disease at the age of, I think, 39, if I remember rightly. So if Adriana Chio, who's a colleague of mine, developed this study of professional soccer players in Italy. They have a risk of developing ALS if they played for up to 5 years, or about 3.5 times the frequency in the general population. If they played for more than 5 years, 15 times the frequency of the general population, which really is a tremendously high frequency. It's not getting to the level of Guam, but nevertheless, it is still very significant. And midfielders in soccer, as some of you may know, are the people who head the ball back when the goalie kicks off the ball. And those are the ones who have a higher frequency, 3 times the frequency of ALS of all of the other soccer positions. So we have been very interested in what are all of these environmental and lifestyle factors that, excuse me, it's one of those problems about doing things from home. So, but in Guam, the signal was very strong. It was quite clear that there had to be something in the environment, something in the diet that caused the disease. In fact, it turns out that all of these environmental and genetic factors that we're talking about here all have a relatively small effect. And that explains why it's difficult to identify them. And I'll illustrate that by our recent work on pesticides as a risk factor for ALS. So these are a number of pesticides that are widely used in the United States. We were able to look at a population of a cohort of about 8,000 ALS cases and compare them with about 25,000 control cases. And look at their exposure to each of these pesticides. And exposure to high levels of these pesticides produced an increased risk of developing ALS of around about 1.07. It's still pretty small. You know, you can understand why we needed large populations of patients and controls to be able even to identify it. But the fact that it is a risk factor, I think, begins to allow us to understand how all these things interplay. So that brings us to this gene, environment, time interaction. So a number of years ago now, we published a paper that set out what we thought was the underlying way of understanding these neurodegenerative diseases, ALS, Alzheimer's disease, Parkinson's disease, and others, in relation to the genes that the patients have in their makeup, the environmental factors that they're exposed to over their lifetime, and how long they're exposed to it. And this was the summary that we wanted to try to put out that explains, I think, our thoughts about it. So there are a number of environmental factors, lead, pesticides, head injuries, and so forth, that are risk factors for developing ALS. There are a number of genetic factors that predispose individuals, probably interacting with specific environmental factors, to give rise to damage to the neuronal cell system that we're talking about. Now, because there are very similar environmental factors and genetic factors that underlie not only ALS, but also Alzheimer's disease and Parkinson's disease and frontotemporal dementia, it seems likely that the underlying genetic basis of the individuals in the population leads them, if they get this environmental exposure and have this genetic predisposition here, leads them to develop one of these diseases, either ALS or Alzheimer's disease or whatever. So that's the hypothesis, and that's where our research is going. We are collecting genetic details, we're collecting environmental factor exposures, we're doing collections of biomarkers of the substances in the brain and in the bodies of these patients. But here's our conclusion from this research providing, I think, what we believe is the current situation with regard to understanding what's the underlying cause and causes of ALS. It's a constellation of clinical symptoms and signs that result from degeneration predominantly of the upper and lower motor neuron. It results from chronic interaction between one or more environmental factors to which an individual is exposed over their lifetime with the underlying genetic makeup of that individual. It's therefore a syndrome without a single cause, and hence, and that's unfortunately the zinger here, without a single treatment. It's unlikely, or it is likely, that once triggered by the specific gene-environment interaction, then degenerative biochemical pathways come in and start to lead to progressive degeneration. So it is likely that treatments directed at the degenerative biochemical pathways can slow the disease, but unfortunately not cure it. And it is likely that efforts to cure ALS will have to be directed towards preventing by either identifying and preventing exposure to these causative environmental factors, or by identifying genetic factors in members of the general population and preventing them from exposure to these relevant environmental exposures. It's an enormous task, and this is, I think, the complexity of that has been what has led to a very slow progress in terms of developing a complete understanding, and also then these treatments that obviously we're all desperately looking for. So that, I trust, did not go too much over time, and that we've got time for questions.
Well, thank you, Dr. Bradley. That was great. I'm going to have you actually unshare your screen for me just so that we can be able to see everyone when we're doing question time. There we go. Wonderful. My name is Sarah. I'm one of the other Everything ALS team members, and I, along with James, will be presenting the questions. We had a lot emailed to us directly last couple weeks, so it'll be good. And if anyone else has questions, you're more than welcome to type them into the chat. So our first question is, what are your thoughts, and do you know of any correlations, but maybe between tick-borne illnesses and ALS? Between
tick-borne illnesses?
Yes, sir.
So it's got to be now 30 odd years ago that there was a flurry of papers and reports of patients having chronic Lyme disease. And there was therefore a period of time when ALS patients were given antibiotics intravenous for long chronic periods to try to remove this believed chronic Lyme disease. In the end, I think that the evidence eventually said the frequency of these antibodies for Lyme disease was no difference in ALS patients than it was in control patients. And therefore, this has dropped off the radar for being a risk factor for ALS.
Wonderful. Thank you.
Dr. Bradley, my name is James. I'll be asking the questions as well, along with Sarah. The next question, thank you for the presentation. It's very informative. Can you explain the possible correlation between high-stress jobs and environments and ALS? And do you believe that high stress could be a contributing factor to the onset of ALS?
Well, you know, high stress has obviously got so many different factors that it produces, hormonal levels in the body, neurological stresses, sleep deprivation, you know, many different things. And we're back into the same sort of mix that I've been talking to you here about. The sports, for instance, high competing athletes are certainly subjecting themselves to stress. And it took an enormous amount of time, research of very large populations of professional athletes to try eventually to make the linkage that professional sports were associated with a slight increase in ALS risk. And it's probably the same situation with regard to the stress at work, the sort of things you're thinking about. It hasn't been demonstrated. It really hasn't been particularly researched at all. But I have little doubt that it will be one of the factors that will certainly increase your susceptibility to some of these other factors that are responsible.
Wonderful. Throughout your presentation, you spoke a lot about cyanobacteria and other environmental exposures, and we're definitely having a lot of questions in the chat. Is there any potential connections between maybe different industry job-related exposures? Some people are asking about specific maybe military exposures or solvents that you may know of.
Yes, those have been what the epidemiological research has tied up as a risk factor for ALS and for these neurodegenerative diseases. So that, for instance, the military, that took a lot of hard work and looking at many predominantly men who've been deployed all the way back into the First World War to be able to recognize a link which was probably about 1.5 times the frequency of ALS in military who were deployed compared with those who were not deployed. And we saw that in the Gulf War and so forth. But it's not an enormous risk, but it is true. Similarly, in terms of what you were asking about work and a variety of different occupations, yes, there have been fairly solid correlations between the type of work that individuals perform, be it exposure to pesticides in the agricultural industry, be it exposure to a variety of different solvents and so forth that contain elements that are neurotoxic. So yes, different occupations can certainly be a risk factor. That's not to say that we should stop every application of pesticides or stop people working in industries which have a lot of lubricating oil and things like that. But it is something to take into account so that we do minimize the exposure that people in those industries have, taking account of all this research.
To kind of piggyback on the military aspect, we had a question in the chat wondering what your thoughts on specifically Agent Orange and the Vietnam veterans were in correlation with ALS as well.
That's a very interesting and still a very difficult topic, because I was lucky enough to go on a tourist visit to Vietnam and see some of the pictures of severely deformed kids whose parents were exposed to Agent Orange, and certainly the primary dioxin in Agent Orange, that toxin, is certainly neurotoxic, but a lot of the research was relatively equivocal. It was not easy to get a certainty about that was that exposure to Agent Orange or dioxins and so forth was a cause of ALS and other neurodegenerative diseases. So, but I do think that that Agent Orange is probably one of the factors that we should add to the list. It's not there on the firm list that people make in these presentations, but nevertheless, I think your question is correct, that it is in fact likely to be a risk factor.
So another question on maybe potential correlations, but we have a bunch of questions about nicotine and maybe smoking as primary smoking or even as secondhand smoking. Has there been any evidence that shows that this may lead to ALS?
Yes, that's probably the best of the environmental risk factors that have been linked to ALS. It's got the highest odds ratio, which is the way of describing this, of any of the environmental risk factors. Now, the interesting thing is that, so smoking, and it's really primary smoking, not secondary smoke exposure, but smoking is a preventative for Parkinson's disease. Now, That's bizarre. And at this moment, we have no way of understanding why that should be, why the yin yang of ALS and Parkinson's disease in terms of smoking. And the mechanism of what smoking does, well, there are many chemicals, obviously, in cigarette smoke, many particulate matter and toxins and so forth. So there's a lot of things to put your finger on, but smoking as a whole increases the risk of ALS by about twofold.
Wow.
There has been recent research that has suggested that the gender gap of ALS patients is continuing to shrink, with female cases equalizing those of males in recent years. Do you think there is any correlation in terms of environmental risk factors that could explain this?
Yeah, you're absolutely right. I mean, that is indeed one of the things that's coming forward as time goes on. Now, don't forget, this is an age-related disease, and the aging population, women do outlive men, and therefore more women are getting into the risk period than do men, because men drop off the twig earlier. So there's lots of possible ways of explaining it, but it does look as though this 1.5 to 1 ratio is gradually decreasing. And you know, that's a very interesting phenomenon to try to understand.
So up till now, most of these questions have been about potential environmental exposures, but this next question kind of talks about, well, where do we go from here? So given your conclusions from your research, how much of this understanding of the disease and the syndrome kind of dictates how maybe future trials and their results can be evaluated, maybe in regards to how can we form trials to be dependent on these large number of causes?
Yeah, and that's a very good question, and it's one that, for instance, we've taken this research that relates to BMA and cyanobacteria and say, well, this one we understand and do believe that it is a cause of ALS, and let's do the trials to see if, in fact, we can prove that it is effective in patients with ALS who we don't know have any particular exposure to cyanobacteria and BMA. And we have 2 trials in operation at the moment. One trial that's already been completed, 2 others that are a larger number of cases that are ongoing. Both of those 2 trials are in Dartmouth Medical School in New Hampshire. And those are both, one's in ALS and one's in Alzheimer's disease. We have another one that is planned to start in Houston, of L-serine, which, as we've shown in animals, will block the effect of BMA. So this is the type of hope that we can come up with. If we can identify more of the elements in the environmental risk factors, then we might hope that we could design different chemicals that would be able to block those ones. And we've been having some research with Mitsubishi Tanabe to try to identify more of these environmental chemicals and be able to see if from that we can develop a sufficiently high risk factor or risk ratio to be able to lead to the development of new blocking chemicals or something of that nature. So that's, I think, the way that a lot of the research is going on. Another theme of research is going to try to identify a variety of different what are called biomarkers. And I'm sure all of you have heard about biomarkers. There's something that is present in ALS patients that can be followed and used to either determine that this patient has ALS, or determine how the ALS is progressing, or determine if a drug is producing a benefit. And all of those things are being developed really very rapidly and extensively. We're interested in that field as well. We've got one group of agents in the bloodstream of ALS patients that we think is a biomarker, and we're trying to look at that to see if that will help us do one of those three things that I've explained. So these are the ways that, that industry and research laboratories are going in terms of trying to develop treatments, as well as what I explained was attempts to prevent exposure to something that might be toxic.
You could not have perfectly segued into this next question, which is there any solid evidence yet for preventative measures for those who may be a gene carrier for the familial version of ALS, such things as to avoid that may potentially trigger an earlier onset, or perhaps dietary supplements to offset potential triggers?
Yes, and there are a small number of centers that are working on the familial forms, and particularly individual genes, and trying to identify what those gene mutations produce and how those might interact with environmental factors. The only factor that up till now is really known in the vast majority of these mutations for familial ALS is time. That is, you don't get it at birth, you don't get it for the first 25 or 35 years of your life, and then suddenly it comes. And so time is obviously a very significant factor there, but we can't change time. So that's the conundrum. Not too many people are working on the environmental genetic interactions on familial ALS. We certainly have been working and are working now on trying to do the gene environment interactions on sporadic ALS. But there are too few familial ALS cases, except in the centers that have really concentrated those patients, to really look at ways of being able to alter and treat or prevent those familial cases developing.
Well, that perfectly segues into our next question as well. What can patients and advocacy groups do to help push this research forward to maybe be utilized in clinical trials or in other studies?
Well, the standard answer, I'm sure you've heard that many times, is money. If we could persuade Congress to provide more money for NIH and the other agencies that fund research in ALS, and the CDC has been, and the CDC has the ALS TDR, that is the organization that looks after the ALS registry. They have been very good and funded our research for the last 5 years. And if more money went to the CDC and to, as I said, to NIH, then more money would come to ALS research. That's certainly the main thing that advocates can do. Fundraising by the ALS community, and that means the ALS Association, that means the MDA, those things can obviously help also a great deal. I mean, there was an enormous burst of research that came out of the Ice Bucket Challenge, and that brought in, what was it, $150 million or something of that nature. I mean, a tremendous inpouring of funds that could be and were devoted to research. We need another Ice Bucket Challenge.
No disagreement there. I just wish someone could think of one to do. In regards to your presentation specifically, if the degenerative biochemical pathways are similar in all ALS cases, then wouldn't a single solution be in the repair of those pathways? At least, do you think that could be a possible single solution?
Well, that has been largely what has been the pathway of drug trials now for the last 15 years or something like that. They've taken, for instance, oxidant, the damage produced by free radicals and oxidative damage. And so the trials have looked at treating with antioxidants and a variety of other things, more specifically directed to the elements of the oxidative pathway. And that's been done in all of these neurodegenerative diseases. Now, none of them, unfortunately, have been have produced the Holy Grail. None of them have produced the final answer for these diseases. And that's to some extent why I think those of us who have been working in this field for a long time have become somewhat jaded, somewhat pessimistic about the fact that these non-specific, if you like, ones directed at the various degenerative pathways are going, in fact, to give us a good answer. There's still all of those various pharmaceutical agents and so forth have produced been quite difficult to produce an answer. You know, riluzole, I mean, I know you know riluzole. Riluzole produced something of the order of 15 to 25% slowing down of the progression of the disease, but it does not prevent people dying, unfortunately, or by and large, it does not prevent people dying. And the same goes for the other various agents that are being developed or have been developed for ALS. So this is the difficulty of, yes, we think that in fact there are common pathways. Yes, we think that we can identify the individual biochemical changes, and we think we can change some of those things. And yet the patient's progression doesn't change a dramatic amount. It's a very difficult field. I mean, it's obviously difficult enough for you who suffer from the disease, but it's extremely difficult for those of us who've been fighting this battle for 50 years.
All right, we have one more question for you. It goes back to your research with the BMAA. Do you know of any way that people can get tested for their BMAA levels or to see if they were exposed?
Well, that's difficult, because the only way at this moment that we know where we can see whether patients have accumulated BMAA is to look at their brains after they've died. And nobody's going to want to volunteer for that, I'm afraid. So at this present time, we've done a lot of research trying to find BMAA in the various proteins, in the blood, in the muscle, in the hair, and in the CSF, in the cerebrospinal fluid. And none of those have turned up the amount of BMAA that we need to be able to measure, as we can measure in the brains of patients who've died with ALS. And so at the moment, no, we don't. That's one of the reasons for biomarker research, is that if you can find something that is a signal for one or other of these environmental factors that can be linked to the disease, then you've got something to work with.
Which I think is a great place to end on the Q&A here, because I know your time is very valuable and you're going to be doing a lot more research for everybody here. I just wanted to thank you once again for joining us. We do go on now to our open forum section. You are more than welcome to stay, but by all means, if you have something else to do, you're free to go as well. Just thank you for the time you have given us. With that being said, I'm going to turn that over to McFinn.
I'm delighted to stay on for a bit. So
more than welcome. We'll be here for a bit as well.
Okay, good.
Yeah, thank
Well,
you.
there you have it, folks. Brought right to your living room. Dr. Bradley, all of us were informed, educated, and we have a little bit of better idea of where this possibly came from to affect us all. Myself personally, I was saturated in lead as a baby and had it for most of my life. And so when I got ALS, that's when I got chelation therapy and found out why I had all those problems during my life. So to each of you, there's an answer somewhere, and none of us are going to give up until we find it. So Dr. Bradley, thank you for giving us a little punch in the arm, a little detective work. And so who tonight out there feels like they ran into some toxic environment and believes that's why they got their ALS? What do you say, Dor?
Well, when I was in Afghanistan, I had to go out to the bomb sites, and I can smell the fumes and where the IEDs were. And that was in 2011. I didn't start getting muscle twitching until about 2017. So I don't know if they were related or not.
Well, it sounds like things that we store inside of us, and for some reason there's a trigger. And my trigger, I really believe, was stress, because I had a big breakdown, and I went to tell my wife that I couldn't handle what I was doing any longer. And 30 days from that day, I lost the grip in my hand, and then it went up my arms, and then it went up my legs. So I believe stress is a big trigger.
Well, the other thing is I played football in high school and college and had a lot of head butts. And so that can be it as well.
Okay, well, there's always there's an answer for everybody. It's like, what are we going to do to find it?
Hey, I have one question while we have Dr. Bradley still on the call that I found very interesting. This person asks, I have sporadic ALS and an identical twin sister who does not. I did genetic testing about 2 years ago for the almost 20 genes known at that time. They were all negative, but I still worry that she might develop it. What are your thoughts on this? I find that very interesting, since twins share the same. I mean, my mom's an identical twin. They share the same DNA. So I'd love to hear what you have to say on that.
That's fascinating. And your brother, in fact, did have ALS, right?
My younger brother did, yes.
Right. So twins have been a very important piece of working out genes that are responsible for a variety of different diseases, because if it occurs in a pair of identical twins, then if you can just search their genome, then you're going to find the gene that is responsible. The research has gone the other way around, of trying to see what you have, that is discordant identical twins. You haven't got the disease, you have a sister who has the disease, right? And trying to find what's the difference in your genetic makeup compared with hers offers the opportunity to try to understand what's the cause of the difference, what's the cause of the disease in her compared with you. That research is, it's a tool which is used, and it really requires much deeper genetic geneticists who are able to do this research than just being able to look at the 20 common genes that you can look at for familial ALS, unfortunately.
Well,
So with all the genes being tested and being negative, I guess her fear is her sister will get it, because she says, I didn't have any of these genes that
right.
popped.
I mean, it's when you have somebody in your family, one's never free of the disease, of the fear, unfortunately.
Yeah.
And even, in fact, if you have a co-worker who has it, it raises your anxiety. Can't help really a great deal in terms of more than what you've done. You've done what you should do. If you should watch NIH because and their websites, because from time to time they put out calls for twins in terms of twin studies, and you and your sister would be very valuable to those sort of studies that NIH is doing.
Thank you very much.
Yeah.
Glenn, what do you have to tell us tonight?
Well, I just wanted to go back to what you were talking about, possibly exposures to anything in my past. And when I was a kid, they used to spray chemical in the air in New Jersey off the back of a truck to fight mosquitoes. And
Sure.
I don't. Then it was DDT. It was either DDT or some insecticide. So I've been wondering if that's a correlation back even to my youth of why I might have. I mean, we used to run through the stuff, so through this fog that they sprayed. So I wonder if that's a correlation of why I've gotten ALS or could have been.
Well,
I totally remember my brothers and I running through the mosquito fog.
Uh-huh.
That was something all the children in New Jersey did.
It was a big thing. Yep.
Yeah.
That's what I did in my neighborhood also. I ran behind the little Jeep that sprayed out the mosquito killer.
Yep. Yeah. I just didn't know if this many years later that would still be a correlation.
Well,
That's sort of the reason that we looked and are looking at the various pesticides. DDT is certainly been shown to be a chronic toxin. Certainly it's not good for your metabolism, so that's why it was taken off the market. But trying to tie that down in an individual is extraordinarily difficult.
thank you. Suitor,
thank
you have a question for us?
Yeah.
Suitor, you're on mute. You'll have to take your, up in the right-hand corner, you can unmute yourself.
Might be Rob can go.
Rob, you want to go next? Rob? You got to take yourself off mute. Can't, there you go.
Just unmuting myself here. Yeah, I noticed lead and mercury listed as possible causes of ALS. So I wondered what Dr. Bradley thought of removing amalgams from your mouth, as well as chelation treatments. Those are not particularly liked by mainstream neurologists, and I can't figure out why. Mine says absolutely not. He said he's never seen a single paper that would document that this has anything to do with ALS, and he's quite adamant about that. So that's one part of my question. The second part is I've noticed a huge difference in the results of a standard blood test, which is the way they test you for heavy metals at most major hospitals and doctor's office, and a forced urine test. When I did a forced urine test, I came out very high on mercury, lead, and nickel, but my regular blood test showed normal amounts. So I wondered if you trusted forced urine tests.
So let me
It's a
answer.
two-part question.
Yeah, let me answer that, Rob, that second question, because an enormous number of the commercial labs that do these tests advertise them to the patients and test your blood and your urine and so forth. Their results are really not reliable. The only reliable tests are ones that the FDA has approved, that NIH and hospitals have performed and are standardized. And it's those that one can believe in. So, but let me then go to the question with regard to lead and mercury. So yes, the association between exposure to lead in a variety of different formats
What's
is...
that lead pencil? Has that got lead in it?
Sorry, Dr. Bradley, go ahead.
So I don't know whether you were saying about lead pencil, but lead pencil actually is graphite. It's not got lead in it. But the association, I think, is really very strong. There are a good number of papers that make the association, different exposures and so forth. But the trials that have been undertaken of chelation therapy in patients with ALS have turned out a significant amount of lead in their bodies, because bones store lead, but have not produced any difference in terms of the progression of their ALS. And that's that's a disappointing feature about it. That has led us to begin to believe that unless you start to do the treatment before the symptoms start, you'll never get any effect. If the symptoms already started, then you're on the cascade downwards, and all the things of past being able to turn them around. And the same goes for Mercury. So unfortunately, the gold standard is treat and see if it makes a difference, but you really have to treat early in the disease, as early as possible.
Thank you very much.
Shooter.
Dr. Bradley, is Radicava, in your opinion, effective in slowing the disease progression?
But that's what the trials that were undertaken in Japan and in the United States showed a slight reduction in the rate of progression, and I think the mortality. So yes, and that's why the FDA gave marketing permission to Mitsubishi Tanabe, because the trials did show a positive benefit. Again, it's a little to the same extent as riluzole. It's not a cure, but it is a slowing down. And if I had it, yes, I would be taking both of those.
Okay.
Thank you. Hamed, you're on
McFinn,
mute.
McFinn, before we jump off real quick, while we still have the majority of people here, I know Deb wanted to say something about our speech project, so if you could give her the
Beautiful.
opportunity to go ahead and to do so.
Great, Deb.
Hi there, everyone. Am I on? Okay.
Yes, you are.
Okay, great. I just want to thank everybody for participating as usual. And I just wanted to give you a couple messages. One, we're really moving forward in the speech project. And what I find very exciting is with the control subjects, people who are not diagnosed with ALS, we're getting so many people out that are not in our community, people who have no connection to ALS, but who hear about our speech project and really want to participate. So I want those people who are on our call right now to know you have a lot of people fighting for you, and they're so moved by this study. And we're doing really great. We're halfway to our goal. And so if there's anything you can do further than what you've already done to help join forces, enlist friends, family members, book club members, church groups, temple groups, mosque groups, athletic groups, anything you can think of. Because once people know about this and how exciting this particular study is and how we're growing it, how we're not having 40 or 50 participants, we're having 1,000 participants. That's our goal. And this is the kind of stuff that's going to help really push the needle for us. So I want to thank every single one of you who have participated, who have opted to do that. I want to thank every one of you who have, referred someone to us. And if we can all join our forces, and if everybody on the call today could actually ask one more person to join our study, either diagnosed or not diagnosed, it would be really wonderful. Just one more person, and we'll get that much further. So you go to EverythingALS, click on the research button, and say, I'm interested in this study. And I just want to thank you all out there again, and I want you to know that we're all in this together. And I don't mean that just as like platitude, but we really all are in this together. Thank you.
And I wanted to do a shout out for Dave Warnick and Bevan. They've been fantastic. They probably referred 20 people last week. And so we are moving forward. We have 500 people who have signed the consent. And we've done this in 5 months, never been done before. So our goal is to have 1,000 by August. So this is, all of us doing it together. So if each one of you, like Deb said, can refer 1 or 2 people, that would be great. We'll get
Thank you. Thank you to everybody.
Hi, Diane.
Right. I think it's a lot easier now that you have the guideline. There's a little person and it tells you, more information. I really like it that way.
Yeah.
Thank
so we
you.
have an avatar. Yes.
Diane, we'll
Ahmed.
have to get you to do a little video for us to help us recruit people. Seriously, I'll contact you on that.
It's good. It's really good. Thank you.
You and your little pooch there, you're going to be our advertising.
Thank you. Ahmed, are you there? How about Anthony? Anthony, you want to take yourself
Thank you,
off mute?
Jamie from ALS Hope. You're always so supportive. Thank you there. We see your comment.
Well, how about
Yeah,
Mark?
hi,
Oh.
this is Anthony. Thanks a lot for your call. I just wanted to thank you, doctor, by the way. You mentioned about stress, and could you tell us why there isn't or hasn't been a lot of research in the past or seemingly going forward on stress related as a risk factor, if that's at all possible? Thank you.